Hi, my name is Houcemeddine. I'm a postdoctoral fellow at Sydney Brenner Institute for Molecular Bioscience, a research institute which is part of the University of the Witwatersrand in South Africa. I'm presenting at the ISMB virtual conference, and my talk is about SWAAT, a new bioinformatics workflow applied for genome-level annotation of variants in ADME genes. Interests in precision medicine are ascending as a medical model of choice in the treatment of many diseases like cancer, neurodegenerative diseases, and cardiovascular diseases. Precision medicine includes pharmacogenomics, which analyzes how the genetic makeup of an individual affects his response to a drug. ADME genes have been the focus of pharmacogenomic studies mainly for their important functional features in determining the fate of a drug in the organism, more precisely its pharmacokinetic properties. There are more than 300 ADME genes. For example, CYP2D6 alone is thought to be responsible for the metabolization process of 25% of the available drugs in the market. ADME genes are highly variable. A recent study that we have conducted on whole genome sequencing data from sub-Saharan African populations has shown that most of the variants with a likely impact on ADME protein function are specific to clusters of populations that share the same geographical space. Additionally, the largest proportion of these variants are rare. This highlights the presence of a genetic diversity bottleneck for precision medicine, requiring a balance between variants useful for population-based applications and between the potential impact posed by variants unique to the individual. The genetic diversity bottleneck suggests that rare variants are important in determining the effect of a drug pharmacokinetics. Thus, understanding the impact of variants at the genome level scale on putative protein-drug interaction is critical for functional predictions in pharmacogenomic analysis. On the other hand, sequence-based annotation only offers a narrow perspective for interpreting the ADME variants. To annotate the ADME gene variants based on protein structure analysis, we have developed the structural workflow for annotating ADME gene targets, SWAAT. In doing so, we kept in mind 4 principles to build our tool. First, SWAAT must have the ability to predict the variant effect on the folding energy and the effect on the conformational behavior of the protein. SWAAT also should be able to tell if a variant is a possible interactor with a drug. SWAAT should integrate different sources of information to judge the impact of a variant. And finally, SWAAT should be easily adapted to other problems of similar nature. For example, you will be able to use different sets of genes that you want to annotate. SWAAT is composed of an auxiliary workflow that is used to prepare an internal database that generates multiple files used either to map the genomic and proteomic data or to provide source files for the annotation process. The second component of SWAAT is the main workflow, which calculates different structural properties and predicts the effect of a variant. SWAAT runs different external programs, but also a large set of in-house Python and R scripts that operate together within Nextflow, a workflow management system. The auxiliary workflow was developed to allow versatility in SWAAT. The user does not have to run this workflow if he wants to annotate the default list of genes of the internal database. These correspond to 28 ADME core genes among 32 others. Core genes are known to be involved in the drug processing mechanism with a high degree of confidence. The auxiliary workflow should be used if one wants to apply SWAAT for another set of genes. To run the auxiliary workflow, you need to provide the PDB files and the list of UniProt IDs corresponding to the genes of interest. In order to run the main workflow, a user needs only the VCF file. All the other dependencies are stored in the internal database, which is generated using the auxiliary workflow. SWAAT uses foldx and income to calculate the effect on the folding energy and the entropy. It also collects different properties that serve as input for a random forest machine learning model that predicts the effect of the variant. Lastly, the report consists of a detailed CSV file and an interactive HTML file for a better readability of the results. The machine learning model consists of 15 features, which were built based on a dataset of 1,491 mutated structures. The overall accuracy is 71%, although also we obtain a high recall value of 92% for variants with high impact. And by high impact, we mean variants that either stabilize or destabilize the protein structure. Here we show also the importance of each of the features and the predictive capacity of the model. and we noticed the importance of the entropy calculation as well as the implication of other sequence-related features. Finally, we are currently expanding the list of the genes to include others with a potential contribution in the drug processing mechanism. And second, we are currently testing the capacity of the workflow to annotate variants with functional impact. The benchmarking dataset is constructed mainly from FarmVar and other data collected from the literature. For more detail about SWAAT, please check the poster number 245. And if you are interested in using SWAAT, you can find the beta version in the following GitHub repository, or you can contact me and I will be happy to assist you. I would like to thank George and Scott for their contribution to this work, as well as my team members at Sidney Brenner Institute for Molecular Bioscience. This work is part of an extended international collaboration under the umbrella of H3 Africa Initiative. And thank you.